So today we have Dr. David Byrne. He comes to us from the horticulture department, if y'all don't know him. Dr. David Byrne did his undergraduate research at Rutgers in strawberry cytogenetics. Then he went on to get his PhD at Cornell in plant breeding and biometry. He did some of his research in Cali, Colombia on mite resistance and cassava. And then he went to the International Plant Research Institute in 1980 and was there until 1983, where he also did some more cassava breeding.
Place.
Where is that? That was in San Carlos,
Okay.
California. Actually, it was a biotech startup,
Okay.
which went broke. That's why I'm here.
So after they went broke, he came back to Texas A&M in 1983 and has been here with us since. And we're grateful for him today to speak to us.
Well, good to be here. Guatemala, that's what I'm going to talk about, right? Just wanted to mention we're having a study abroad in Guatemala this summer, so if your professors will let you go, we will be glad to have you for 3 weeks in July and show you lowlands up to highlands and everything in between in Guatemala, just a great diversity of agriculture and ecosystems and cultures. That's all I have to say about that. Then we get into rose breeding, which is exciting too. I was asked, Wednesday or Thursday to give this talk. So this is sort of a modified talk I gave at an international conference, slightly modified so it would be more pertinent to this group. So I'm going to talk a little about rose breeding and genetics research in North America, just sort of gloss over that, but I guess really, let me see, I got to figure this out. There I go. So what I'm going to do, and just to give you an idea of what's going on, I looked at rose breeding in North America, looking at private breeding, public breeding, and major trends. And then I'll talk a little about the rose testing programs we have out there. And then, of course, rose research at the various places throughout the U.S., the public research. And the first question I had is, who is breeding roses? I know I am, but there's a lot of people out there. It's really surprising. I had various sources of information. I went to patent for the last 3 or 4 years, and there was about 14 people releasing things. So 14 breeders, companies, what have you. Not all U.S.-based. A lot were European as well. If you look at, there's another database called Help Me Find Roses that has all the registrations for roses. And there's about 40 breeders involved in that, and they don't protect their stuff. They release it and promote it and what have you. And then I did a survey, which was really interesting. It was an online survey, and I sent it out and got 1,800 responses or so. And 135 of the responders said they breed roses. This is in North America. And I'm there, wow, that's a lot of rose breeders. I know maybe 10. So there's a lot of people I don't know out there. Most of them are small programs. They do it for fun a lot of times. We'll talk a little about that. It's a whole different ball game compared to corn breeding. And you see this in ornamental breeding in general. Where are the breeders? If you look at the breeders that patent, you see a lot of them are in California. And then there's some out here that are beginning to release. They haven't had too many releases yet. But the majority have come out of California. And of course, California nice and dry, and it's a really nice weather. And a lot of those are not well adapted to east of the Rockies because of our humidity. And so there's really a big shift in rose breeding over the last 10 years. If you look at the number of patents, about 78 per year, but it's gone down quite a bit last couple. These are 3-year means, just so I didn't have as many bars there. But you can see it pretty steady through here, and then it's coming down. So we're down to about 40 in 2010 to 2012. So it's really come down considerably. If you look at the patents by North American breeders, about 32. So about half the patents for roses in the U.S. are bred by U.S.-based, or U.S. and North American, both Canadian and U.S. Based breeders. And the others are, there's about 4 major breeders in Europe that send a lot of the materials out here. They have their representatives, they send the materials, and they patent them. and market them. So Cords, Tantau, Polson, whatever. So there's various there. So a lot of, at least half are being produced by other places. If you look at the roses registered, you can see, again, a lot in California and the West Coast, but you're getting more in the East Coast, just small breeders. I'm among that too, and so there's at least one other breeder in Texas. Actually, when I looked at the data on the survey, there's like 9 or so, which really is interesting. But anyway, so again, there's a concentration out here in the West Coast. If you look at the numbers released, West Coast is probably the highest numbers released in roses. And so if you look at registrations over the past few years, about 96 roses are registered every year. Okay, so it varies quite a bit. And remember, there's 32 patented, so fully 2/3 are not protected at all.
Where are they registered?
They're registered at the American Rose Society have a register of roses. And this would be an example of one of those people that did this sort of thing. And a lot of them are nurserymen that breed on the side, and they release roses for their production only. So they come out with unique things every year. And Ralph Moore, who donated a lot of his materials to our program, he did a lot of this in the last 20 years of his life. He would release 5 to 10 roses per year that were different, so the people who really liked his roses, they would buy them. So he just keep putting new products out there is basically what he did. And there's various other breeders or nurserymen that do it. The Antique Rose Emporium does this as well. So they have things, they register them, but they don't patent them. And then you look at the survey response, and this is a lot more, but again, if there's just singled out, that means there's one person there, or one breeding program there. But again, there's quite a few here, but then you start looking at east of the Rockies. There's a lot more small breeders east of the Rockies. Okay, and look, there's 9 in Texas. I think I know of 2 of them, so I gotta get to know a few more of these small breeders. But there's a lot of interesting work that they do that they don't report. And actually, there's a Rose Hybridizers Association, and I work with them trying to sort of answering questions for them and helping them in some of their research. But right now we're trying to figure out exactly what all these small breeders are doing. Some are using very unique species in their breeding. So they're not thinking of releasing things for money, they're releasing things because they like doing what they do. So it's a very different approach. If you look at the age distribution of the rose breeders, because in our survey we took data on demographics too, you can see a lot of them are in their 60s, in 50s, and of course 70s and 80s. And if you look at this a little more carefully, fully, I think it was 40% of the rose breeders were retired, and they were doing it in their retirement. Okay, so they're not commercial breeders. They're people that are very serious hobbyists, by the way. I don't know if you've ever gone to a rose meeting, you'll see how serious people are about roses. It gets to the fanatics sometimes, which is interesting environment to live in. And but anyways, you can see a lot of the breeders are 50 and older in the survey anyways. If you look at where they're coming from, again, a lot, almost 40% retired, but they're coming from a wide range of backgrounds. There's actually some breeders among them too, which is nice. But artists, communications, education, engineer, financial, industries, health, quite a few horticulturists, and these are usually the nurserymen or somebody dealing with propagation and selling of plants that they do some breeding on the side and then release through their organization. Some lawyers, various management types, research, and students, but again, the bulk are retired people who breed roses because they like breeding roses. So what about the trends in rose types? I don't know if you're familiar with the various rose types, but of course one is miniatures. And Ralph Moore, who I mentioned before and who donated a lot of his material to Texas A&M, was known as the father of the miniature rose. And he released, maybe 300 different miniatures throughout his breeding career, which spanned 70 years or so. He died at the age of 102, 103. And he bred till the day he died, really. He lived on the nursery, and that's what he would do. And you can see that the miniatures, early on in the early 90s, 50% of everything that was released or was patented was a miniature rose. And that's when the industry of small potted rose plants came really popular. And they started selling a lot of potted rose plants, and they needed miniatures to fit those pots. And so that's why there were so many released at that time. Since then, they've had enough varieties, but there's still releases. Hybrid teas, which is the typical rose that you always think of when you talk about roses. And that's where people really are fanatical about their hybrid teas and showing roses. And you go to a rose show and you see a large room just full of displays of roses, and sometimes it's a single stem, and they rate the single stem on perfection. Sort of like a dog show, but with roses. And so if it's not exactly how it's supposed to be, it's out. There's one little thing, and there's all sorts of ways to get them to the show that make them perfect. And it was just fascinating to listen to some of these presentations when I went to the American Rose Society meetings. But you can see this is the average over all these years, about 25, but you can see the last 5 or 6 years have been a lot less hybrid teas released. Hybrid teas are known to be susceptible generally to a lot of disease and pest problems because they're not really bred with that in mind. Okay? If you look at the floribundas, and it's just sort of a smaller shrub type thing with a lot of flowers, flora, and you can see that's gone up and down, but it's generally on the upswing now. Generally, they do have a little more resistance to various diseases, better adapted plants than hybrid teas. And then you look at the shrubs. This is the one that's really gaining popularity. And I think sometimes whether it's a shrub or not depends on the marketer. So some of those hybrid teas that have resistance are probably shrubs now. And so you get that sort of shift in the marketing, because these are marketing names really. And they're named on their phenotypes, not on the genetics or anything. And so it tends to be marketing. There's a lot of other different types, but I'm just going to talk about those 4 to give you an idea. So if you look at, here I have the patented roses we just looked at. This would be 2010 to 13. So hybrid teas about 10%, miniatures maybe 12. Floribundas and shrubs, 25 to 30% of the patented roses. And there's some other classes that aren't in this grouping. But if you look at the registered roses, hybrid teas are still 30%. Now a lot of these people are breeding roses so they can display them and compete with them. So this is where you get into a different mindset. And they will spray them, they will finesse their plants so you get a perfect plant. And so that's where a lot of this is coming from that perspective. And otherwise, miniatures, floribundas, and shrubs, 10 to 15% or so. Very different what's coming out, these two groups of breeders. And actually, just so you know, the patented people are in here as well, okay? Because I didn't separate them. So the patented people also register their roses. About trends with black spot resistance. This, of course, one of my emphasis on research. And in that same survey, we asked about what traits were important. And again, this had 1,400 responses, but we got a total of 1,800. This was a summary before we got all the rest of them. But you can see when they rate them, the only one that's very important on average is disease resistance. And of course, fragrance. Everybody wants a fragrant rose because that's what you think about with a rose. If you get a cut rose, not so much because they're not fragrant. But if you're talking about garden roses, fragrance is very important. Of course, color and number of flowers, productivity, plant size, and going down and down. Prickles, not terribly important. Which surprised me. I always figured ones without prickles would be very important. But it is if everything else is good. And then that comes in. But if it's a choice between one with prickles and a wonderful flower, lots of flowers, and one without prickles that doesn't quite have that flower, they'll take the one with prickles. Okay, so it's sort of how you rank things in your mind and make those decisions here. The other thing I did, I thought, well, how do I get a sense on how breeders are reacting to this? So what I did, I went through the patent database, and I said, okay, I'm going to search on rose and the word black spot and see how it changes over the years. And you can see here, in 1992, 30% of the patents had both words in it. In 2013, over 70%. So it's changed so that at least the sensitivity to the importance of black spot has changed tremendously. Now, are they resistant to black spot? Well, not necessarily. When you look at a couple trials, and we did a lot of evaluations here, us, and then there was a really good trial out there in Tennessee. And so I put all that data together, and there was 413 roses evaluated. And we evaluated for foliar diseases, so not necessarily just black spot, because we have anthracnose out there as well. And so if it had more than 10% damage, we put it as susceptible and less as resistant. So it's pretty stringent. And when you do that, 6% come out as resistant. So we still got a ways to go. But the newer varieties tend to have a little higher level of resistance to a lot of the diseases that are common. So we're getting there, but we're not there yet. So I got a job for a little while. The other thing that's changed a lot in roses, in the testing of roses, previously we had the AARS, which is All-American Rose Selection. And this was a national thing. They would put those roses out, and they would spray the dickens out of them to keep them healthy so they could evaluate them. And so it was a heavily sprayed test. Well, that sort of collapsed about 5-6 years ago because all of a sudden they would promote these things and they would die because of disease, and nobody wanted to spray them in the gardens. Not nobody, but not enough people. And so we got a shift. And so the first thing, about 10, 12 years ago, Earthkind rose evaluations. Again, this started in Texas with Steve George up in Dallas. And he selected a whole range of things in Dallas. So he did the first evaluation at the experiment station. He eliminated most of them, and then worked with the growers and nurserymen and put them out throughout Texas for further evaluation. And then after about 10 years of testing, they say, well, these are the best ones, and we'll call these Earthkind and promote them as such. Okay, and so that's what they've done, and done a very good work with that. And so basically it's a no-spray landscape performance trial. So they don't get any sprays at all. So if disease gets them, that's okay, you eliminate. Okay, so this was really a big shift in the paradigm of growing roses. And so it's really caught on. There's now a northern session of Northern Brigade where they're going up to the central states, Minnesota, Wisconsin, Ohio, Illinois, various other places, to evaluate a different set, because the adaptation in Texas doesn't necessarily fit the one up in Minnesota. So they're going through their evaluations now. And then the other thing that's starting, sort of when the AARS collapsed, they realized they needed a no-spray test. And so the American Rose Society stepped in and said, okay, well, let's start one. And this year was the first time they actually planted. And so again, they're going to put no spray trials out there, and anybody can enter. I think you have to pay an entrance fee of $300, $500, supply your plants, and again, it's modeled off a program in Germany where they've done this for years, and it's really quite successful. They have 11 sites in Germany where they evaluate various breeders, their best stuff to see which is really, really good, and then they can promote it that way as ADR winner, etc., and it's a great promotion mechanism here. So what they're going for is regional recommendations, because we realize that no one rose will do great everywhere. I mean, the U.S. is pretty big, a lot of different environments. And so those are the test sites. So they have quite a few test sites. Again, no spray. We have a standard way to evaluate. And then the evaluators will be trained and go through standardized testing, and hopefully things will come out fairly good. Because the AARS, the other thing there was it was very political. And this is hopefully less political, because sometimes the big producers of new varieties would say, well, I need one this year. And so they would get one, whether it was justified or not. So, you know, there's some things going on there too. But anyways, so this is sort of the new approach. Okay, what about rose research? If you look at North America, there's really not a lot of rose research out there. You know, I know all these people. You know, University of Minnesota and Wisconsin, in Arkansas, Washington University, Missouri Botanical Garden, Violin Research Innovation Center in Canada, of course, Texas A&M. So those are basically the players in the rose research world. And there's where we're located, basically east of the Rockies. There might be a little going on west of the Rockies, but nothing on landscape roses, and that's what I was concentrating on. If you look at the public breeding programs, there's 3 of them. And actually the one up in Minnesota is resting at the moment, so they're not doing any rose breeding. So it's really Texas and Canada, which are, you know, how extreme can you get with respect to environments, right? And so Canada, cold hardness, black spot resistance, and that's it. Of course, I do black spot resistance and heat tolerance. And of course, adaptation and, you know, the usual things you want in a good rose. And so those are the sort of things that we're dealing with. And if you look at what Minnesota's done, they have released various things. Very cold, high cold hardness is what they've been focusing on. They still have selections under test, but they're not producing any more seedlings. The people in Violin have a long history of rose breeding. And about 5 years ago, the government closed it down, and then the industry said, wait a second, and so they stepped in and got started again. So they're just getting started. The new breeder in there is just getting his feet wet. He's got some seedlings out there, but they have released various series, like Canadian Explorer series and Parkland series, that have good cold hardness and good landscape qualities. Unfortunately, they don't do well in Texas. Just can't figure this out. Okay, and then, of course, Texas A&M, we do a little breeding as well. The funding is an endowment, and I'm always looking for donations, so I'm a fundraiser as well. Never thought I'd be that way, but yeah, that's okay. We get some state funding, grants, contracts. We're always looking for funding. You know, every place you can get it, do collaborations with private companies. You know, if we want plant materials, there's no problem. They just send it to us here. You kill it if you want to. It's okay. You know, so we get good cooperation that way. They don't send us too many checks yet. I got to work on that. So we're working on that. The objectives: disease resistance, heat tolerance, releases. Well, we're trialing quite a few things out of the Moore collection in the U.S. and actually in Japan as well. And we got some of our stuff that's being trialed as well. We haven't released anything as of this point. The first 10 years that I was in rose breeding, it was mainly research breeding and looking at germplasm and things like that. So it was only recently, since the Moore donation, have I really pushed to get things out as varieties. If you look at rose taxonomy, there's one guy, and actually Walter Lewis, who's out now at the Missouri Botanical Garden, actually began in Texas as a rose researcher. But then he wandered elsewhere. That was in the 50s. But he still always kept his interest in rose, and he's now actually revising the North American species, rose species. So he's going to come out with that. So that's a big step. Roses, from a taxonomical point of view, are extremely confusing, because you have all these species that intercross, and sometimes they're in the same area, so you get hybrids all over the place. So it's very different from a lot of crops. So you get a lot of interspecifics here. Rose viruses, you know, if it wasn't for these guys, we wouldn't have as many viruses, I think. They keep finding more. And especially Ben Lockhart in Minnesota, he's found 3 or 4 in the last 5 years. I don't know where he's finding these things. Hopefully they aren't in Texas. Giannis in University of Arkansas is working really with respect to roses, on rose rosette, which is a major problem that's arising in the East Coast and actually all the way down to the Midwest in Oklahoma. We're beginning to see it in Texas. This virus all it does is kill your plant. And there's, once it's in there, you can't really get it out. So the only approach you have is prevent it from getting in there. It's transmitted by a little mite, an aerophoid mite that just floats in the air and transmits very nicely. And so that's what he's doing, and that's good. We're trying to learn more about this particular virus. Of course, disease resistance, that's one of my loves. I love seeing disease in rose gardens, because that's when I know something's susceptible or not. This is black spot, which is a major disease around. There's been a lot of work in the University of Minnesota, University of Wisconsin. They work together, and they did a lot of the initial work for evaluation and breeding. They looked at single gene resistance and identified RDR3 and found a few markers. They started working on partial resistance, which is probably my highest interest, is in the partial resistance to black, or horizontal resistance, as is normally called. And of course, continue to look at molecular markers and what have you to try to find easier ways to handle things. They've done some work in Ploidium rosa, and of course male sterility in Rosa satidra, which is a native species, which is, there's female and male types. So it's dioecious in many cases, which is fairly unusual for a rose. And then we do some stuff here at A&M. Now, just to give you a background, I think a couple years ago I probably talked about this too. I came here not as a rose breeder, but as a peach breeder, which I'm still a peach breeder. But in the early 90s, this gentleman, Dr. Basie, came up to me and he said, hey, he asked me to count some chromosomes. He had a little tube of shoot tips, and in a previous life when I worked on strawberries and did cytogenetics, I knew how to count chromosomes. And so I said, well, yeah, I can do that. And then, so I did, and he was happy, and he donated money to the university, and so we started a rose program, which is totally unexpected in my plan of life. But that was okay. It's an exciting crop. It's really quite interesting. And then later, about 5 years ago, Ralph Moore came to me and said, would you like a few roses? I'd love to donate something to your program. And what he wanted to do was donate all his germplasm to the program, which he did. And so we have all of Ralph Moore's germplasm, the stuff that survives, because it was developed in California, so it doesn't have a lot of black spot resistance. So we have to spray it to keep alive. Some doesn't have heat tolerance. We can't do much about that. It dies. So it survives in California, but not in Texas. But Dr. Basie all he was interested is trying to get a really healthy plant to hang those wonderful flowers on. And he was one of these backyard breeders, very serious about it. He was a mathematician working at Texas A&M. He taught things like imaginary numbers and things like that, which is way beyond me. But when he was off work, he'd be in his backyard breeding roses. And he did a lot of interesting things with ploidy levels and stuff like that. So he did a lot of interesting things that I continued with. And then in 2007, of course, Ralph Moore comes up and gives me all his stuff. And his emphasis was horticultural qualities. And he had such a wide range of material, it was just incredible. And so now we're beginning to work with that. This is his 100th year celebration. And he was also featured in several large rose magazines. Again, known as the father of the miniature rose. And his idea was continue with education, research, and sustainable rose production. And if you look at Ralph Moore and things he released, this is sort of gives you, when he started in the 30s and into the 2000s, but every decade he was releasing in his prime about 100 varieties, so 10 varieties a year. Tremendous number of varieties he was releasing. And I'll just give you an idea of some of these. The miniature roses, stripes and halo roses, moss-crested, brachiata hybrid, rugosa hybrids. We won't go into the details here. Just we got a lot of variability, a lot of diversity to work with. And of course, now they both Dr. Basie and Ralph Moore knew each other. They never met each other, which was interesting. But anyways, they said, well, now we need to unlock the treasures of the entire rose genus. One man and one lifetime are not enough. The logical answer is a university where the torch can be passed. So that's sort of the idea of these guys. They wanted to continue with stuff. And so they gave it to A&M to do so. And so the vision was, of course, a center of excellence for rose and ornamental breeding. Because Ralph Moore did more than just roses. He dabbled in a lot of ornamentals as well. And of course, concerning, involving a lot of different things. So the rose breeding program here is for well-adapted to landscape roses, so disease-resistant, heat-tolerant, high number of flowers, and excellent horticultural traits. And of course, that's never easy. The ongoing breeding program, actually, many of the commercial roses are tetraploids, but there's quite a few diploids. So actually, I have a diploid and a tetraploid breeding program, and then I, of course, pass them back and forth as well. But on the diploid level, you have a lot of species that have really high tolerance to all sorts of environmental issues. And on the tetraploid level, you have really superior horticultural characteristics. So you really have to work with both. But I rather work on a diploid level because it's a little quicker. You don't have to work with 4 sets of things, just 2 sets. So I want to do as much on the diploid level as possible with respect to integration, what have you. And one species we worked a lot with, or have worked a lot with, was Churiana, trying to get resistance to black spot and heat tolerance into ever-blooming background, which would be on the diploid level as well, because most of the species bloom once a year. Of course, most commercial varieties bloom every 4 to 6 weeks. Okay, so they're ever-blooming. Of course, we have a tetraploid level breeding program and integration. Of course, we're combining all sorts of these characteristics together. We do a lot of evaluation. I work with Brent Pemberton in Overton because they have much better black spot than I do. Just beautiful disease out there. And so it's a good place to be if you want to evaluate things for disease. Again, evaluations, I just want to point, this is the trial we ran. And of course you get things like this that do well and things like this after 2 years that are dead. And so you get a wide range of adaptability among the roses. And again, maybe 5% were really well adapted and the others were somewhat well adapted to dead. And this dead one is actually Fragrant Cloud, which is just a wonderfully fragrant rose. But unfortunately it doesn't survive. But when I crossed it with some of Basie's material, highly resistant, this is what I got. So it's something that has much better adaptability. Beautiful fragrance. Unfortunately, only has a few flowers, so it's not quite there. But you can get the fragrance into resistant material. I don't think that's a problem. It just takes a couple, 3, 4, 5, 20 generations, whatever. It takes a while. So we've developed, things that are very floriferous and healthy, large flowers. This is a fragrant one. And with Churiana, we've worked a lot with on the diploid level because it has very high heat tolerance and very high resistance to black spot. And this would be a horizontal resistance rather than a single gene resistance. And we can distinguish. It's got some problems with powdery mildew, but we can handle that. So from that, we've developed things like this. Again, very floriferous, no prickles, which is nice because I bleed a lot in the rose field, as my students will attest to. And using stuff like this that's fairly nice, cross it with better horticultural qualities, we can make a lot of progress because this has very high resistance to black spot and heat. If you look at the breeding population we're using for breeding, actually for genetic studies, we have a whole range of things out in the field at the moment. Again, a lot of it's based on Churiana at this point, though we're beginning to incorporate other species as well. They also include chinensis, which is the source of the ever-blooming trait. There's commercial chinensis clones that we use. And so they have some fairly decent horticultural qualities. We also have incorporated 97.7 population from a colleague in Germany. It has multiflora, and the problem with it is it's very heat sensitive. So it's a little hard to incorporate, but we're working with it because it gives us a lot of segregation for heat tolerance, and that's going to help us try to figure out what genes are important here.
How many genes control the ever-blooming trait?
There's one loci. It's a recessive gene.
Okay. Can it be cloned?
They're working on it.
Okay.
They're working on
And
it.
is
There's a good
GMO
French.
Is GMO accepted in technology accepted in roses?
Well, it's funny. I've done surveys when I talk to groups, rose groups, and I ask them, well, would you grow a GMO plant and start discussions and what have you? And if it's for disease resistance, 90% say yes, and 10% say no. There's always 10% say no, no matter what, right? But 90%, most of them would. I mean, if it was for some other trait, they wouldn't be so excited about it. Now, the ever-blooming trait, it'd be nice to get some of the species ever-blooming right away, but if not, it takes me a couple of generations. So I can do that relatively easy on the diploid level. Some other people have done the crosses and go once blooming and pop it up to tetraploid level, but that just, the numbers you have to go through to make it ever-blooming just so much higher. And so I do it on diploid level and then pop it up.
Okay.
But in these populations, we get a wide range of things segregating. We're beginning to do all the phenotyping now. And, of course, we're going to be doing the genotyping as well. We'll talk about that a little. Disease resistance work, we've been working for a while. Tiffany's here in the audience. She does a lot of this work. She has stacks and stacks of Tupperware things to do her disease analysis or evaluation. But again, there's two types, race-specific and horizontal. A lot of our populations are looking at horizontal resistance, so we're keeping track of the race-specific, the dominant genes there too. We're looking at genetic studies. Of course, marker trait associations is where we're hoping to go so we can evaluate parents and make sure we make the right crosses to combine all the right genes. Again, collaborations with lots of people on campus, off campus, at Dallas, Overton, Germany, out there in Minnesota. So we work with a lot of people. And again, on partial or horizontal resistance, depending how, I don't know why in the rose literature they call it partial. I've always used the horizontal. I remember I used it in a grant proposal and they said partial, why go for partial resistance? Oh, okay, we used the wrong word. It's horizontal. It can be very high. You can get to a point with Turiana where you see a lesion, you got to look for lesions. So, but it starts but doesn't go anyplace. Anyway, so basically looking at genes that limit the growth of the fungus is what, and as far as we can figure out, we've tried various pathogenic races. It's pretty effective overall pathogenic races, and that's what we're looking for. We want a fairly broad resistance that's not going to break down easily. And of course, this is what she does, is evaluate resistance as a detached leaf assay. And she's done hundreds of these, thousands probably, right? And she's still going. We just bought 300 more containers for her to work with. So we got a few things to look at. Heat tolerance is another thing we work with. It's Akkard Gravestein who works on this. Of course, heat affects a lot of different things on these plants. And of course, Texas is pretty good with heat. And so, again, collaborating with a lot of people. And you look at just the difference in heat with respect to flower size and color. This is a common variety called Knockout. And this is Belinda's Dream. Again, heat stress here. And this actually was, I think, a 3-hour heat stress. So you can see that if you time it right, it makes a tremendous effect. Of course, I think it was at 45 degrees centigrade too, but it was 3 hours. Again, abortion of the flowers. And of course, this is just to give you an idea of how hot it got, 3 seasons. I guess this in 2011, when on our site, well, here in College Station, I guess it's in August, the mean maximum was 38 something. so 102, 3, and the minimum was somewhere about 80, so it was very, very warm. Roses do much better in cooler climates. And so what we've done over the years is actually go through, and we do evaluation 3 times a year, as the previous graph actually showed, was spring, the heat of the summer, this would be August, and then in fall, so usually late October, November, we'll be out in the field evaluating these things. With respect to the productivity and what percent of the plant's covered with flowers, just sort of a quick rating tool that we can use. And of course, if they don't produce flowers, they're not terribly useful. And there's some that do really well in the springtime and then crash in the summer. And so we looked at a whole range of things, and this is just average flower intensity, 5 being good. So we want at least a 5 or so. So there's not many that on average do 50% of the plant covered with flowers. But there's some that do really poor. Of course, those wouldn't be good any year. If you look at the poor ones, well, they're poor all year round, pretty predictable. But then you have these, where in the springtime they have a lot of flowers, the fall time they have a lot of flowers, summer they look almost dead. It's amazing the differences you pick up. So this is very heat sensitive, and there's some varieties we work with here to look at things. And then you get these things that it doesn't matter. If it's 100 degrees, it just keeps producing flowers. Flowers can be a little smaller, sometimes a lot smaller, but they always produce the flowers, where the others just stop growing. They just don't do anything. And so we have good germplasm that had heat tolerance, maybe 5% of what we have out there. And some of them actually have nice flowers, which is even better. So the future challenges, of course, drought and salt tolerance. We've done some work there in collaboration with a graduate student, Amy, Kai, Jinhua Nu, in El Paso, Terry Starman here at College Station, of course, Brent as well at Overton. And because we're running into, we're going to run into water limitations and poor water quality, especially for landscapes, because they're going to get hit hardest because they're considered least important. And so you're going to have plants that can handle this. So we're beginning to look at some of the variability here. And we're just starting to get our feet wet there. We got a long ways to go, but it's something we need to look at. We've looked a lot started looking at interploidy crosses, again, in collaboration with George and David over there in the cytogenetics lab. And of course, this is Jake who works with it. Looking at diploid, triploid, tetraploid roses, we've got the whole range of things. And so the question is, well, how do we integrate between ploidies? And how do we do this? What do we get when we make these inter-specific crosses? Most breeders are interploidy crosses. Most breeders have no idea what ploidy level they're working at. They say if the cross works, fine, and that's all they worry about. Well, I want to know a little more. And so we've done a lot of crosses. Of course, we looked at pollen size, and we found in our collection, the Moore collection, 14% were diploid, 8% triploid, and the majority were tetraploid. And so, well, how useful are these interploidy crosses? So we did a bunch. Of course, you look at the pollen, you see a range of pollen sizes. And just guessing, this is maybe a triploid, diploid, and a haploid pollen from a triploid rose. So maybe, if you do a triploid rose, cross it with a diploid, you might get diploids, triploids, and tetraploids. So, interesting. Let's see what the possibilities are here. So we did crosses, diploid by diploid, diploid by triploid, tetraploid by triploid, and tetraploid by tetraploid. And here's the fruit set. So a little less on the triploid crosses, it's expected. And we only use one triploid in this work, so it's home run, which is a specific triploid. And the results may differ with triploids. I fully expect them to. If you do germination, now you get almost 20% with the diploid by diploid, 25% with the tetraploid by tetraploid. Of course, these are less, especially with the diploid by triploid, only 5%. So you've got to make a lot of crosses to get a few seedlings out. So it's an expensive thing to do. it's a lot more productive to do the tetraploid by triploid cross because your germination is still pretty good. So what happens? So we did this. We did these crosses. We put the seedlings out, and the question was, well, what seedlings are we actually getting? And so this is just some preliminary data we have when we cross the tetraploid. In this case, Golden Gardens by home run. We get 63% triploid seedlings, so we're getting a haploid pollen grain from the triploid, and 38% diploid, tetraploid seedlings, so we're getting a tetraploid. So we're getting haploid and diploid pollen grains coming out of that triploid. We didn't see any triploid pollen grains coming to the seedlings anyways. So either they didn't survive or they don't really get that far. And if you do diploid by triploid cross, we get 45% diploid seedlings and 54% triploid seedlings. So it's almost about half and half if you look at both things. And we're looking at other progenies now, and we'll get better numbers here.
Which one was a female?
The female was always the diploid and the tetraploid. So it's the triploid is going to be the male. Because they really, I mean, just the number of gametes they produce is fairly low. So we have problems with the number of pollen grains, and it's same going to run into a number of female gametes. So always use the pollen, which is going to be the triploid. Okay? So it's interesting that you can get some diploid seedlings out of a diploid by triploid cross, because one of my interests would be to take your tetraploid traits and get them on the diploid level so I can breed on the diploid level. So it may be a way, expensive, but it might be useful. And of course we've done molecular marker work. Who hasn't? You can't get away from it at the university, right? You got to do molecular marker work. So we have, and we've had a lot of people work on this. And we've done several things. And one was just look at germplasm diversity and answer the question, what is a rose? And of course, the answer is a multi-species complex. It's a conglomeration of 7 to 10 different species. It's really a mess. But it's an exciting mess, if you can understand that. If you look at domestication, it's domesticated, of course, in 2 places. You get the domestication in China and, of course, in Europe. And, of course, when trade started from China to Europe, the Chinese roses went to Europe. Now, the Chinese roses at that time were the only ones that bloomed continuously. The European roses bloomed once, maybe twice if they were really good. And so when the Chinese roses got to Europe, they were really exciting and very expensive too, because only the rich could afford them. But they would plant them in their gardens, they'd let them open pollinate, and people would take seed, and all of a sudden you get all sorts of things coming out. But it's a diploid by tetraploid cross for the most part. And so you get a lot of triploids, but we know triploids throw all sorts of things out there too. So eventually you get tetraploids that are ever blooming. And that's what the basis of the modern commercial rose industry is. And so we've looked at some of the various hypotheses of where roses come from. Just these are some of the basic species that we start with, and we get various types of roses, and don't worry about the details. It gets complex. All sorts of interspecific crosses. You can do the microsatellites and get a cool thing like this, which is really too small to read, so I'm not going to worry about it. But you also find there's lots of sports out there. You know, single gene mutations. We found Old Blush is a variety that's been around since 17-something. And they're clonally propagated so you can maintain these over the centuries. And it has all sorts of sports. You get the green rose, you get Roulettii, which is a compact form. You get Arctic Charles, which is a little darker and more petals, and there's 5 or 6 others that we sort of identify. And there have been some work on this saying, oh yeah, this might be, but it very definitely seems to be. And so lots of different things there. Now, of course, we've done mapping. Who hasn't? Again, everybody has to do mapping. That's the thing to do. Now, I personally find my map kind of boring, but that's okay if it gets me to where I want to go. But we've done various maps, again, collaboration with Germany, Holland, and France, and here with tetraploids with Clemson and Israel. So we made diploids and tetraploid maps, and now we're combining them to try to understand what's going on. And here's that exciting map. I don't see anyone excited out there. All right. But the exciting part is if you can find a trait that's related to a marker you can use in breeding. And so that's really the exciting part. And so that's what we're trying to do. If it can facilitate selection, if we get some markers to do that, I'll be happy. The maps themselves are great publications. But, you know, again, the traits we're interested in that we're characterizing, phenotyping, adaptation, and floral and plant traits. Now, we've seen a lot of advances in marker and data technology. And, of course, we've done, I started here with isozymes and rapids and AFLPs and microsatellites, and now we're snipping away. And, of course, it's digital genotyping now, all that sort of stuff. It's all exciting because you can get the genotyping done, if we can work out the details, that is, and figure out how to analyze it. Little details, but that's okay. We can do that. And, of course, we collaborate with lots of people, various graduate students here. Some are not listed, actually. And Marco Bink, and I couldn't remember the other guy's name, so he's off. But he's there, and he helps a lot. They're a group out of Holland. Now, I'm also involved in the Rose Breed Project, which is on fruit, because I work on fruit. And I'm trying to do the same sort of thing on roses, and basically getting standard phenotyping going, and then learn how to genotype, and we're getting there. And, of course, do the QTL analysis, but not just with one family, but with a group of families. And there's some software being developed out of Holland called FlexQTL, which uses a range of different families to sort of put it all together and find your QTLs from a standard map and what have you. So that's sort of where we're going with this. Again, with black spot and heat tolerance and plant architecture, productivity, flower types, a lot of different things we'll be phenotyping. We'll genotype all those and then run them through a program and magic comes out. Well, maybe not. But we'll figure it out if it does. And of course, this is not possible without a lot of people. I've been blessed over the years to have some great graduate students and a lot of visiting scientists who sort of add to the program and help us. Most of them are pictured here, though I'm missing Sarah Jones and, of course, some of our student workers, Daniel, Kyle, and Jessica, which without them, the plants would be dead. And so we got a lot of help getting this all done. And we got a long ways to go, but it's been a fun trip. If you have any questions, I'll be glad to answer them.
You haven't mentioned rootstock.
Don't have to.
Well, are most roses grafted?
No, actually not. They're going away from the grafting. There is some rootstock work done, but I don't do it. When we breed, we also select for ease of rootability. And so the trend now is to self-root them and put them out there. So we're getting away from rootstock from a commercial point of view. A couple reasons for that. If you use a rootstock, it's a 2-year cycle to propagate your plant. If you do a cutting, it's a 1-year cycle. So it's a much cheaper approach. And it seems to work pretty well in most soils. We'll probably run into some problems in specific soils, no doubt.
How long does it take to breed a new
rose?
rose? Yeah.
Well,
The time you make the cross.
from the time you make the cross to release, probably about 10 years. 5 to 10 years, depending on who you have to help push the evaluation, because the last 5 is the final evaluation stuff. If you do it in more sites, you can make a decision quicker. Yes?
How do you give them heat treatment? Like, do you put them in growth chamber or just?
Well, mostly we just put them out in the field here in Texas. But we do have a growth chamber as well, so we can heat up that growth chamber and give them heat treatment there as well.
How much temperature can heat tolerant variety give?
We do a heat shock, or we did a heat shock at 45 degrees for 3 hours, and heat tolerant does fine, and the susceptibles don't do so good. But we can, when I showed you the first pictures of the knockout, which had the red flower and the pink flower, that was, I think, at 36 degrees with 28 at night. So it was fairly warm, but not incredibly warm. Yeah.
And most roses will self-pollinate?
Depends. Yes and no. Not most, no. A lot, on the tetraploid level, a lot will be able to self-pollinate. Diploid level, it varies quite a bit. That said, the fertility on the tetraploid level varies extremely, from essentially zero to very good. When I was in Germany, I was talking to breeders and before, and the first thing they tell me is that make sure you select your parents well. And they didn't mean for characteristics, they meant for germination. Because that's a big problem. There's a lot that just don't produce much seed.
So there's a rose out there that's male sterile,
Yes, the Septidra.
and you anticipate using that in your breeding program, or?
Yeah, actually, I do. I haven't used it yet. There's been some work with it up in Minnesota and Wisconsin. The interest in that, it's actually called the prairie rose. It's native to the U.S. And the interest is it looks like it has resistance to rose rosette virus, which we don't have much sources of resistance. So it'll be 4 or 5 generations till we get to a commercial type from that.
You use the term patenting. Is that the same as what we call PVP?
No. It's actually, you actually patent a clonal plant.
Okay.
So you patent the genetics of the one plant that you can clonally propagate. So yeah, it's similar but different.
Okay.
So you patent fruit trees and potatoes.
How expensive is it?
It's about $3,000.
That's
per
about
patent.
the same as the PVP.
Yeah.
And maybe that's the reason why you don't see a lot of patents, you see a lot of registrations.
Yeah, I think so, because a lot of times it's very local, regional selling.
Okay.
And so they might sell, 20,000 plants, so it's hardly worth it to patent.
That's right.
Yeah, and so they just produce more things every year, so people need to get the newest thing.
Sure.
Yes,
The Japanese have developed a blue rose by genetic manipulation.
yes, they
Do
have.
they have anything about that?
Well, it turns out that it's not a bright blue. It's sort of a light. So they tell me it's not. I haven't seen it. I've seen pictures of it. It's not real bright blue. But yeah, they put a gene from what was it? Delphinium in there, and they got a bluish rose. Yeah.
What is the future of it? Is it moving? I mean, how?
I haven't. What they were trying to do is develop it as a cut rose flower because they wanted to control it. They didn't want it in landscape quite yet. Because if you talk to rose breeders, where the large rose breeding companies make money is off cut rose flowers because they can control that pattern very well. Where you get into a landscape situation, for about 5 years you make money and then it just sort of gets propagated and it's hard to keep track of it. And so the idea was to put it into a cut rose flower, and I haven't seen any in the market, so I don't know how far they are in there. But they have done it, and it's been through all the registration loopholes, hoops, not loopholes, hoops in Japan anyways. Any other questions? Yes.
You mentioned the horizontal resistance to black spot. How many races are there currently?
Well, they have identified 11 races.
So it overcomes the resistance fairly.
Yeah, well, we tried. We only tested against 2 or 3 that we actually had the individual races for. But yeah, it seems to be pretty good for those 2, 3.
Do one more question.
Is fragrance is qualitative trait or you take 5 to 6 generations?
Depends what fragrance you want to talk about. There's like 100 different fragrances in roses too, which makes it confusing. There is one fragrance where there is a major gene involved, but there's other minor genes with respect to the amount, how it gets out, and it's complicated. But there are a few major genes involved that they've worked on.
All right. Thanks everybody for coming today. Let's thank our speaker.
Before the graduate students leave, I have one request of you. The original planning for October 25th, November 1st, were you guys practice runs on your ASA presentations. Most of those were changed to posters. So the proposal for me to you is you all want to have a poster competition here on one of those dates. And I'm more than happy to give both a small cash prize, and we can also have some sort of an award or certificate for the best poster to put on your CV. Okay, so if you all want to do that, please see Laura get on the sign-up sheet for those dates. If we get enough signed up, we'll go ahead and have a poster competition. All right? Thank you.
All right.